Good morning, everybody, and welcome to the session on poultry meat processing. This is part of the educational program through the Midwest Poultry Federation. Just as a couple of housekeeping things, I would like to draw your attention to the bottom of the screen where there's a Q&A tab. If you have questions as we go along the presentations, please feel free to type your questions in there. We will be monitoring that question on the Q&A chat window and pose those questions to the speakers as we go along. We are very excited to have some good speakers lined up for this session, and hopefully that will generate some questions and increase interest amongst you guys. Our first speaker for this morning is Dr. Harshavardhan Thippareddi. He's a professor of poultry science at the University of Georgia, and his research area is in the specific area of poultry processing and food safety. This morning, he will be talking to us about value-added poultry products with a focus on white striping. With that, I'm going to turn it over to Dr. Thippareddi.
Good morning, Manpreet, and good morning, everyone. As Manpreet mentioned, I work here at University of Georgia, and it's a pleasure to be speaking this morning and presenting some of the work that we have been doing at the University of Georgia in collaboration with the Meat Science program here in the Animal and Dairy Sciences Department, and also the USDA ARS folks right here in Athens. This work is a collaboration between all the three entities, and although I'm presenting, that represents all the work that has been done. We'll be talking about utilization of breast meat with myopathies. And currently, most of this breast meat that has the myopathies has not been utilized properly and has been discarded or sent to products that have little value, mostly to rendering. And we wanted to find ways we can utilize it or make it more palatable and provide it to the consumers, and also add value to the processors and improve the profitability. So here is an outline that I'll be going through this morning. I'll talk briefly about some breast myopathies, how we currently use the breast meat, different whole muscle products or methods of improving, and muscle tissue changes that occur with these breast myopathies, and then get into further processing, how we can enhance the value of these products or ameliorate the deteriorative effects or detrimental effects of these myopathies on the breast meat consumption or to the consumer, including moisture enhancement, blade tenderization, and also preparation of comminuted products such as patties or fresh sausage, and eventually the emulsified products. And then we'll conclude at that point. If we look at the breast myopathies, the three main types of breast myopathies that have been out there reported widely or extensively are the woody breast, the white striping, and spaghetti meat. Each myopathy is a little distinct, and the manifestations in terms of the product quality or palatability is different. But one thing that we need to keep in mind is that all of these are not mutually exclusive. That means if one breast fillet has woody breast, that doesn't mean it cannot have white striping or it doesn't have spaghetti meat. Some of these breast fillets can have one or more of these myopathies, and thus utilization becomes a problem. Marketing of these breast meat with these myopathies of as intact muscle or meat can be an issue, and it has been an issue. And several of the processors currently separate this meat or separate these fillets, and some of them have found value in making other products, and some basically send it to rendering. So think about a breast meat with a pound, $1 or more value, going to rendering with 6 cents per pound. You're losing almost like $1 in value for these products or these breast fillets with the myopathies, and the loss in economic value is significant. In some of these meat processing operations, especially for the large birds, processors have been seeing anywhere from 20 to 40% of the breast meat or breast fillets that have the woody breast or other myopathies included. So the loss in economic value is significant for these processors. So there is a need to find alternate value or alternate ways of improving the value of these products. When you utilize these products as whole muscle products. Sorry. Yeah.
Real quick, Harsha, can you advance your slides from the live view with the yellow arrow on the right of it? Thank you.
Oh, I'm sorry. Yeah. Is that visible now, better, Manpreet?
Okay. The live view window, the yellow arrow on the right side.
Okay, there. So marketing these products or these breast fillets as whole muscle products is harder, but there are some unit operations that we can do to improve the eating quality of these products, such as blade tenderization, injection marination. But further processed products are probably the better option for it. So what are some of these products that can be prepared? You can grind the meat and market it as a ground chicken or chicken patties. Currently, the market for ground chicken is almost like $1.50 to $2 per pound. And when you grind, you may lose some functionality to it in terms of water holding capacity and like that. But still, you can, with the increase in value, you can maintain or maintain the economic value of the product. You can make them into fresh chicken sausages. These are getting to be more popular on the market today. These are primarily coarse ground product with some spices and minimal ingredients such as salt, probably not phosphate. But still, if you look at the standards of identity for chicken sausages, there has not been one out there in the CFR. So it is possible to add some phosphate to these products and increase the water holding capacity. And eventually you can use these breast fillets as emulsified products such as hot dogs or bologna or similar products as well. What are some of the muscle tissue changes that we see in some of these myopathies? Here you can see the histological section of normal, which is the top, and the woody breast meat. In the normal breast meat, you see polygonal muscle fibers that are tightly packed and relatively uniform in size, whereas in the bottom section, you can see the woody breast meat, the histological section. You can see that the number of muscle fibers are reduced significantly, and also the muscle fibers are of different size and shape. And some of them are loosely or more organized connective tissue with mild inflammatory cell infiltration. So with these changes, you see significant loss in water holding capacity, especially at the sections where you see the woodiness in the product. In this slide, you can see the histological sections of normal, which is represented by N, woody white striping, which is WS, SM is spaghetti meat, and WS or /SM is white striping spaghetti meat. And you have both the longitudinal sections as well as the cross-sectional view of those histological sections of the meat. With the normal, you see regular polygonal profile, compact and normal shaped muzzle fibers. Peri and endometrial spaces are normal, whereas with white striping, you see abundant endometrial fatty tissue infiltration and also fatty infiltration in some of the fibrillar part as well. And in the spaghetti meat, the muzzle fibers are reduced in number and also spaced apart. So you actually see these fibers falling apart visually when you look at the spaghetti meat. And also in white striping or spaghetti meat combination of myopathy, you see significant and abundant proliferation of endometrial and perimetrial connective tissue, and also split fibers and significant adipose infiltration. So these changes in the connective tissue is what is evident when you look at the meat and also look at the quality of the meat in terms of palatability. Here you can look at the characteristics in terms of the white striped and spaghetti meat, as well as the combination of white stripe and spaghetti meat in terms of the weight. And also, if you look at the bottom three rows, the top height, the middle height, and the bottom height, these are significantly different between normal white striped and spaghetti meat. And also, the top height is more evident with white stripe and spaghetti meat having greater height compared to the bottom portion of it. So even within the same woody breast or white striped or spaghetti meat breast fillet, it's not uniform. And also you can see gradual decline towards the bone portion of the breast meat or breast fillet. So it's more evident, these myopathies are more evident towards the top portion, the skin side, with general decline towards going towards the bone side. Okay. Here you can look at the approximate composition of the white striped and spaghetti meat. As we have seen, the moisture is much different, especially for the white striped and spaghetti meat. Protein is lower, as well as if you look at the collagen, the superficial, it's significantly higher in the white striped breast fillets and spaghetti meat breast fillets as well. Here you can look at the muzzle characteristics in terms of the water holding capacity or the T2 spectra that shows the difference between, even in a normal breast fillet, the top portion, the surface part of it, as well as you go to the interior, which shows the bound water, the myofibrillar protons, and also the extra myofibrillar protons from the spectrum. When you look at the intensity, or relative intensity and the T2 characteristics of these, you can see the significant differences between the white striped and spaghetti meat. And if you were to have the same analysis for the woody breast, they'll be similar to the white striped and spaghetti meat, but a little more pronounced in those characteristics. So what impact do these have, breast myopathies have, in terms of the palatability and the characteristics of the meat? Number one, the approximate composition of the fillets is significantly different. It has higher moisture, higher fat, and also higher collagen content, which translates into lower palatability and hardness of the muzzle itself, whether it's raw or cooked, and also lower protein content. the protein content may be lower, but if you look at when you make a product, when you comminute, that may even out, even if there is a little bit lower protein content compared to a whole fillet. So you can use some of this meat as an ingredient in further processed products. In terms of the functionality of the muscle proteins, you see a lower water binding capacity, and we're not sure the ability to form stable emulsions and gelation properties, but the evidence we have so far from doing some of these studies shows that it's not significantly impacted when you combine some of these breast fillets with myopathy and proportionately add to normal breast fillets and make them into sausages, fresh sausages, or comminuted emulsified sausages like hot dogs that we will see as we go along. In terms of the intra-fillet variation in the myopathic lesions, you see differences between cranial versus caudal ends of the fillets, as well as superficial versus the deep portions of the fillets. It's more pronounced at the superficial side of the skin side of the fillet compared to the deep portions of the fillets. And some of the processors have been slicing the breast fillet horizontally so that they can utilize the bottom portion or the bone side of the breast fillet as a normal breast fillet and discard the superficial part of it or the skin part of it, skin side part of the breast fillets. In terms of the visual characteristics, you see a significant outbulging in shape. In some cases, you see hemorrhages and viscous fluid. But USDA FSIS requires that if there are any hemorrhages, they need to be removed. And if the hemorrhages are extensive, they need to be, they cannot be used for human consumption. So in terms of further processing, what can we do to enhance the palatability of these products? The first thing we looked at was moisture enhancement. This work was done at the ARS with Dr. Bowker and Zhang and others. Look at marination composition, salt and phosphate, injection and vacuum tumbling, as well as vacuum tumble marination. The impact of woody breast, lower marinade uptake, only about 53 to 69% of the normal uptake. So you lose significant water holding capacity of the meat when you use the woody breast fillets as intact fillets. The water retention, retention of the moisture or the marinade is about 71 to 85% of the normal breast fillet. So even though you pump or inject a lot more of the marinade, it's not held. It's lost as purge over time. And also even in cook loss, after cooking, you lose higher moisture as cook loss in woody breast compared to the normal breast fillets. So the next slide, we can look at the impact of using woody breast on the marination. Marination improves the woody breast shear force. And in terms of the texture profile analysis, increase in hardness is observed, as well as springiness and other characteristics. And you can see the difference between normal and severe woody breast. And if you were to have moderate woody breast, it will fall somewhere in between. What does moisture enhancement or injection, marination, and vacuum tumbling do? On the left, you have non-marinated fillets. When you cook, what happens? And on the right, you have the marinated fillets. Most of the other characteristics, such as the hardness, cohesiveness, fibrousness, and springiness, etc., are increased with woody breast fillets. And also the juiciness is lower in woody breast fillets, in non-marinated fillets. Whereas when you have the marinated fillets, the juiciness increases to the same level as the normal breast fillet. So the juiciness part of it will be very similar to a normal breast fillet that is cooked, injected and cooked, compared to a woody breast fillet. But the other characteristics, such as the hardness, springiness, and like that, remain the same, or the woody breast will still have higher values for those characteristics. If you look at the skin side and the bone side of the portion of the fillets, you can see the difference between that. Bone side, it's very similar to the non-woody breast or normal woody breast, whereas on the skin side, you see significant differences in non-marinated, but those differences are reduced in marinated fillets. So marination does enhance the juiciness of the product and also reduce a little bit the hardness of the product as well. So the next unit operation we looked at was blade tenderization. This work was done in collaboration with the USDA ARS here in Athens. Blade tenderization is used extensively in the beef side of the meat industry to make the meat cuts similar in quality, as the variability is significant for meat steaks. So it's applied for subprimals and then cut into steaks. So we looked at blade tenderization as a unit operation to enhance the palatability of the woody breast meat. Here we looked at the normal, moderate, and severe woody breasts, and then both on the raw side and the cooked portions of the woody breast, we had a control and blade tenderized product as well. When we looked at the blade tenderization decreased the compression and shear values in both raw and cooked woody breast fillets compared to the untreated controls. So blade tenderization did increase the quality or improve the palatability of the product. And the question now is, can you combine that with marination or injection and see if the juiciness and other characteristics can be improved? The next product that we will look at is the comminuted products. Here, looking at the chicken patties, where the meat is ground and then made into patties and cooked. As you can see, the quality can be different between normal and woody breast, and also the degree of woody breast can make a significant impact both on the fillets as well as the patties. The hardness and juiciness, the main characteristic that is important for palatability, is very similar when you convert that into a patty. And also the hardness difference between normal and woody breast is reduced when you make it into a patty form and cook it. Although the springiness, you still see the difference. But if you look at the consumer palatability, it may not be that significant or that evident. The next set of products that we made are comminuted products, which is fresh sausage. We had a normal chicken breast fillets, 0% woody breast, and incorporated different degrees of woody breast meat into the fresh sausage meat, 25%, 50, and 100% woody breast into the normal chicken breast fillets, and the same percentages with the severe woody breast, and looked at the composition as well as the quality of the meat or the sausages. So if you look at the approximate composition of the fresh sausage itself, You can see that the normal breast fillets or sausage made with normal breast fillets, the moisture is lower compared to the moderate and severe woody breast, 76 versus 77, 78%. The protein content was lower as well, with almost like 22 versus 20% or 19% of the protein content. And also the total and insoluble collagen content is significantly different, especially the insoluble collagen. 3.3 milligrams per gram of meat versus 5.6. So if you were to use this meat intact, there would be significant differences. So here you can look at the formulated sausage approximate composition incorporated at different levels, 25, 50, and 100% of moderate and severe woody breast. And even in here, as you go higher on the woody breast content, whether it's moderate or severe, you see higher content of insoluble collagen, higher fat content, lower protein content, and a little bit higher moisture content as well. But you look at the hardness, the texture profile analysis of the fresh sausages, the hardness and cohesiveness, they're very similar, although the product with the woody breast, whether it's moderate or severe, is slightly lower than the normal chicken breast fillet meat used in the fresh sausages. When we look at the cook loss, with higher content of the woody breast, we saw gradually higher cook loss observed in the fresh sausage subsequent to cooking or during cooking. The next iteration of products that we looked at are the emulsified sausages. We looked at different iterations of the hot dogs, initially with the beef with pork fat, beef and pork hot dogs, and then normal chicken breasts hot dogs, and then incorporated with moderate and severe woody breast meat into those hot dogs. So if you look at the texture profile analysis, with the beef and pork, you saw a little bit higher hardness, whereas with the chicken meat, you saw lower hardness, although they're not significantly different from each other. The other characteristics, such as gumminess and chewiness, as well as in the next chart, we'll show the other characteristics, the adhesiveness, etc., are very similar to the sausages or the hot dogs prepared with beef only, beef and pork, or chicken breast fillets that are normal as well. So incorporation of the woody breast fillets into these emulsified sausages probably gives you the best value in terms of amount of meat that you can incorporate, and also minimal differences in characteristics of the product as well. Here we used the thigh meat and looked at the yields of the product. And regardless of how much of the woody breast, severe woody breast we incorporated, the peeling yield as well as the chill yield are very similar to each other. So you can incorporate this meat into products, comminuted products, especially the emulsified products, with minimal loss in functionality. Conclusion, breast meat with myopathies, little value as intact meat today, or you lose significant value. But you can enhance the value of these products using unit operations such as blade tenderization, injection marination, and more importantly, through combination or products that rely on combination, such as fresh sausage or even more emulsified sausages, and increase the value of this meat significantly. With that, I'll stop there, and if you have any questions, we can discuss. Thank you.
Thank you, Harsha. Everyone, if you will please continue to use the question and answer option in the bottom of the symposia window. Then we will deal with those. If we don't answer them verbally, we will send you responses via the chat system. So please make sure you keep utilizing that system. Our next speaker will be Dr. Manpreet Singh from the University of Georgia. Manpreet is an expert in poultry food safety, and he is going to be speaking about hot topics associated with poultry meat safety. Manpreet?
All right, thank you, Dina. Good morning, everybody. So as you can see, the topic for my presentation here is very broad in the sense of hopefully talking about some current or hot topics which we are faced with or challenging us from a food safety perspective in poultry and poultry products. So just to kind of lay the outline for the presentation today, we talk a little bit about the current state of food safety in the poultry industry, and perhaps that will lead us to see where the needs are on how we need to be enhancing the safety of poultry and poultry products, and at the same time, possibly get into some of the ideas of where I believe we're headed in terms of any sort of issues that are related to controlling or addressing the challenges in the safety of poultry and poultry products. So with that being said, I would not be doing justice if I did not kick off this presentation with this classic slide that we always have up there. This is the performance standards that we deal with or we are held accountable for in terms of ensuring the safety of poultry and poultry products. So if you look at it, again, there'll be from a whole bird or a carcass perspective, there's a performance standard for salmonella at about 7.5% and about a 10.5% performance standard expectation for Campylobacter. Mind you, this is on the whole birds. And then you can so on and so forth see for turkey carcasses, comminuted chicken, and then all the way down to chicken parts, which if you look at it from a performance standard perspective, salmonella, the expectation is no more than 8 out of 52 in a moving window for chicken parts, and 4 out of 52 for Campylobacter, which I believe is a significant challenge, personally, to manage that in on the chicken wings or the chicken parts, for that matter, for Campylobacter. That being said, we also know that currently, excuse me, there are categories into which we look at information or we're categorizing plants based only on salmonella performance standards. Mind you, we don't have categorization based on Campylobacter yet, but this is strictly based on the salmonella performance standards. So out of the performance standards for salmonella, you can be classified into category 1, which means you're less than 50% of the performance standard. Category 2, which is you're still within compliance, but you are above 50% of the allowable percent positives. And category 3, which is basically you're exceeding or you're failing the performance standard. We all strive to be in category 1. Or category 2 for that matter, again from a food safety perspective, from a compliance perspective, category 1 and category 2, I don't see a difference. But from a process control perspective, from an academic perspective, or looking at my plant performance, differences in categories, it does project that if I'm a category 1 plant, stricter controls and better management of my hazards. That all being said, when we look at the performance standards and where we're headed or how or where are we today, these next 2 slides will kind of lay the foundation of where we are today. I hope this is clear in your slides. If you look at this slide, basically what you're looking at is a level at which we are from a Campylobacter infection perspective. Your x-axis is the years and the y-axis is the cases per 100,000. The red dotted line down here is where the Healthy People 2020 target was for Campylobacter infections. So we were expected to be at about 8.5 people per 100,000 from an infection perspective. But we are operating at about 20-ish, which is kind of the upward trend. So we're nowhere close to our 2020 Healthy People objective. And I know I was in a meeting where we're now talking about Healthy People 2030, so already we're moving these targets based on the information we have. The goal with the performance standards was to reduce infections. However, here we are, and this is what the current trend is showing from a regulatory perspective or the scientific disease control and prevention perspective. Similarly, if you look at the salmonella infections, again, the red dotted line down here is where we were expected to be, which is at about 11.4 cases per 100,000 people. However, we're operating slightly above 14 cases per 100,000 people. So although we're doing a good job in controlling salmonella, at the same time, it's not reflecting in the reduction in the cases from salmonella. Mind you, this is through food in general, but again, poultry is one of the major food commodities which appears in our top 10 list for causing salmonellosis or campylobacteriosis. So that kind of leads me into where and how or what type of process controls do we have in place for packaging. The first question we always talk about when we're trying to design and look at packaging control programs is where is the process that we focus on? We talk about processing, we can talk about live birds, we can talk about countries, we can talk about consumers, we can talk about retail, but in essence, all inclusive. So as long as it's exclusive of the other, if we want to make sure that we can have a really inclusive process. Even if you look at the process, we're looking at these guys workers, we're looking at starving, and we're also looking at inside out very often as things in addition to the studies. If you look further into the type of products that are cut up and further processing, you can see that those can also be areas where we can be preventing the packaging control. Guys, give me 1 second. I think I'm getting a message on the chat window that there is some issue with some audio, so I'm going to really quick put up my in case you're wondering, I did practice this, and it is working just fine.
Okay, so moving along, as you see that in a poultry processing operation, this is kind of a outline of how we can look at a overview of poultry processing all the way from grow out through processing into packaging. I've shaded 3 areas, which are primarily the scalding, picking, and evisceration, which we believe as being major points where cross-contamination can happen. Therefore, when we look at and design our pathogen control programs, we're really focused on the picking area, at the evisceration area. Scalding, we always have discussions and there's varying opinions, but we're not quite there in saying that at the scalding stage, we should be adding some sort of a antimicrobial for pathogen control. So some of the challenges when you look at pathogen control are, questions that we need to answer. First and foremost, what are our goals for pathogen control? By that I mean, are we trying to reduce prevalence, or are we trying to reduce the concentration, which is the number of pathogens? Yes, the short answer is prevalence, because that is the regulatory requirement. However, from a process control perspective, for me to have a better handle and understanding of my process, I probably would look into the concentration of pathogens as opposed to the prevalence. At the same time, we need to look at what type of systems are we using for processing, and what are the systems we are using for application of antimicrobials. For example, are we using immersion systems like a post-chill dip tank, or are we using post-chill spray bars where we're only spraying the antimicrobial onto the carcasses? So again, with those things, the challenge becomes where is the pathogen located within the product? So is it on the surface, like the skin of the product, or is it in the inside the product, which means underneath the skin layer? And that's where sometimes with parts there could be some challenges as from a control perspective. Other things to think about when we're addressing pathogen control is, does your current system for process control provide you adequate information into the process? This kind of falls back into my first point I made, that if we're going to look at prevalence, that's great. But at the same time, it might not tell us how many organisms, and you might be compliant from a prevalence perspective, but if you're seeding your chiller with a high concentration of organisms, then eventually your prevalence will go up too. So having an understanding of that will give you better insights to your process as well. Other things which are very common we look at are aerobic plate counts, Enterobacteriaceae counts. We perhaps look at those as indicators towards how our pathogens are going to behave for any control program. I would suggest, if that is the route we're going to take, we definitely need to look at what is the correlation of that indicator organism with the actual pathogen. We've done, Dr. Thippareddi in their lab have done some research and looked at Enterobacteriaceae as a possible organism which could correlate with Salmonella and Campylobacter. And historically, we know EB can fairly correlate well with Salmonella, but not as well with Campylobacter. So if you're going to use an indicator organism which you want to look at for Campylobacter control as well, you might want to look at making a right choice with that indicator organisms to add that indicator organism so that you have the correct correlation. So that being said, we know one thing, that we are a growing industry from a processing perspective, and that also means that the increase in consumption is, or the future of the poultry consumption is very dependent on assuring that consumers have a safe product go for consumption. So with that, where are we heading from a control perspective? As I mentioned, it is my belief enumeration is something which we definitely need to look at. We definitely want to research more and understand so that we have a clear perspective on our process capabilities. How can we improve our process based on those pieces of information, or adding new equipment is another thing I want to discuss. And perhaps I'll talk a little bit more about some control strategies and biomapping as well. So from an enumeration perspective, Salmonella enumeration would be really a good option for us to understand the process and get more details and insights about the process. However, if you have tried to do any sort of quantification of Salmonella from field samples, it is very challenging, and there is no reliable method available to accurately quantify. There are still research which is ongoing. There's some products which are being validated in terms of some semi-quantitative methods or some methods which can give you a correlation for a close enough population of Salmonella on the product. Currently, we use the most probable number method, and we use it a lot, but I tell you what, it is very labor intensive. It is fairly expensive to do an MPN method, so it doesn't quite cut it from a high throughput perspective. For Campylobacter, again, enumerations are a key. We do know that there are high concentrations of Campylobacter on the product, so it can be very useful and relatively easy to enumerate Campylobacter. However, we also know that there are inconsistent levels of Campylobacter. These numbers of Campylobacter coming into a poultry processing plant are varying. So it could be that your method, which is there, might be there to detect a certain level of Campylobacter populations. And if you have lower than that, and you might look at your plates and say there's nothing on it, but at the same time, it could be that you're just not being able to detect it because of inconsistent numbers of Campylobacter coming in. From a process improvement perspective, we always heard about chillers, and then we got into pre-chillers, and now we're into post-chillers. So there's so many different segregations of the process that we have gotten into that at each stage we're trying to put in an intervention so that we can decrease the load or the number of organisms on the product. So we're going into pre-chillers where we might have some sort of an antimicrobial, then go into the main chiller where we definitely have some antimicrobial, and then going into a post-chill, which could be even a post-chill immersion tank or sometimes post-chill spray bars where we're trying to add another antimicrobial. I've seen in some places where there is a post-pick dip tank. So right after the picker, the birds are going through a dip tank where antimicrobial can be added. So right when the bird is hot and the leaves have been, sorry, not the leaves, the feathers have been, the feathers have been picked. So that way you're adding an antimicrobial onto the surface of a hot bird. I mentioned retooling of existing antimicrobials. By that, we've been in many conversations, and I've been hearing a lot of discussion on adjusting and using different levels of PAA, different levels of pH for PAA application. We've done significant research on adjusting the pH of the PAA, and I have yet to see data where it shows me that higher the pH, the more effective the PAA. So if there is information out there, I am very interested, or we are very interested here at University of Georgia to have those discussions to understand how those are being attained, because in our research settings, going all the way from a pH 8.2 up to about 11, we are not seeing much differences in at least Campylobacter populations or Salmonella populations being reduced, or any differences in the efficacy of those. A lot of folks are also using CPC and using these drenching cabinets for CPC, which I believe over the past 5 years, there's been a few plants who have switched to that, and those are showing promise as well. The other thing is parts chillers, or we have parts dip tanks where only parts are being dipped into these high concentrations of an antimicrobial to be able to get added efficacy of an antimicrobial. The focus for control strategies that we'll look at is we mainly are trying to focus and saying we're going to add an antimicrobial or multiple stages of our unit operations. So all we're hoping for is an additive effect all the way through the process. Most of these interventions which are being used, they could be spray or immersion based. However, I will say from all the data which is out there that immersion seems to be more effective than sprays, which based on the type of spray system that is being used. Other things we need to know is if the sanitary dressing practices that are ongoing, are they still effective? We don't know, but if you have a baseline, if you have data which is being collected, that'll help you provide insight into how you need to design that. And maybe you have interventions which are already effective. And we hope those continue to be the way they are and you're getting consistent results. The main challenge where we're dealing with is a moving target, which means the number of organisms from Salmonella and Campylobacter coming in is constantly varying, and that is where we might look at variations in efficacies of the antimicrobials. The other thing I want to address and wanted to talk to you about is basically biomapping. This is a very useful tool, as we believe, to provide insights on your process. Basically, it is a systematic evaluation of your process and specifically focusing on microbiological performance of your process. You know your process best. You know your high-risk areas if you're collecting data. So that should give you insights of how you need to be designing a biomap for your process and help you answer the questions that you're going to set forth. The key to having a biomapping is you need to have a very serious conversation amongst your team about what is the goal of having that biomapping process done. Are you looking at the process? Are you trying to understand sanitation or manufacturing process, or you're just going to look at the finished product? And we've had enough conversations about, or had a discussion about, I'm mentioning indicator organisms or pathogens. What are you going to look at? If you're going to look at indicator organisms, fine, but are those correlated, and can you interpret that data and correlate that data to your pathogen performance is something that you will have to look at from that perspective. Here's an example. Dr. Thippareddi had developed this visualization, and I've been able to use that in a lot of my presentations, trying to give you just a snapshot of, a poultry processing operation and looking at the stars, which basically indicate those are the possible places you could be collecting data to get more insight. At a minimum, the shaded areas are where you're possibly applying some sort of a intervention. So in that scenario, you would, at a minimum, want to collect samples from those areas to be able to understand how you are performing and what are your high-risk areas within your operation. So with that, I just want to wrap up with some thoughts that I have on the processing side of food safety. Is I think we all can agree that the microbiological safety of poultry is a significant concern for consumers. And we also agree that Salmonella and Campylobacter are the true primary pathogens which are kind of giving us the headaches of addressing food safety in the poultry industry. The poultry processors also need to evaluate the process controls using indicator organisms, but I should caution that they need to be correlated back to pathogens so that you have a better understanding of the process. And biomapping is, in my opinion, an essential tool, which does give you that insight to your process. Other things to think about: we discuss PAA so much in our intervention strategies. Is that the end all be all? Are we there and we've solved the issue, or of how we need to be using PAA? We've been seeing some information, some plants discussing the issue of resistance of the pathogens to PAA. That is something worthwhile researching, and we should look at that so that we are ahead of the curve and be prepared for those if that situation arises. We all know and we all hear about the effectiveness of PAA against Campylobacter and Salmonella. They're not the same pathogens, they're different, they behave differently, so how can we address them Are we going to be able to use one type of a parameter which can address both issues? And we know that we've moved from a whole carcass to cut up and parts. That's what the consumer demand is. But parts yet are giving us a main challenge. So can we be using high concentrations of PAA or any other antimicrobial for the matter only in the parts, which helps us because that is the ultimate product we're going to manufacture or process, can potentially help us reduce the use of antimicrobials upstream. By that I mean towards at the inside-out bird washer step or the pre-chillers and other phases. With that, that's all I have, and I believe I am caught up on time, so I would be happy to address any questions if there are. Hopefully the audio was solved, Dina.
It was. Thank you. You just had to plug in. So, Manpreet, I have a question for you and Harsha as a tag team. I know y'all can handle being a tag team. Harsha, unmute yourself and turn on your video. Are there any links between food safety and muscle myopathies?
No, those are two different things, Dina. Any product with muscle myopathy has no relationship with foodborne pathogens or food safety issues.
Do we have any spoilage organism concerns, considering that you mentioned that the proximate composition is notably different for the muscle myopathy tissue, in particular that it has more water present? Do we have issues with yeast or with other spoilage organisms differently for the muscle myopathy tissues?
Yeah, the moisture content is a little bit higher in meat from breast fillets with muscle myopathies, but if you look at the water activity, it's going to be pretty similar. So water activity is the one that determines the relative growth of organisms at the same temperature. And so since the water activity is going to be very similar, very high in general, unless you reduce it significantly, it's not going to make an impact. So even though the moisture is higher, even the pH is a little bit higher, but not high enough to make a significant difference in terms of the growth of spoilage as well as foodborne pathogens.
Yeah, and so for Manpreet and Harsha, again, has there been any linkage between on-farm microbial counts and the incidence of muscle myopathy from birds coming out of housing environments with different microbial levels?
Dina, if I may, I am not aware of, and I don't think there is a correlation between microbial counts and myopathies. So and I'll let Harsha speak to that in more detail, but I'm not aware of any of that information.
Yeah, and going back to your question, Dina, is there any evidence or research to show that there's no correlation? To date, I haven't seen any research conducted on the prevalence of foodborne pathogens and the prevalence of muscle myopathies. No, I have not.
Thank you.
Thank you.
Okay, Dina, I think. All right. So with that, thank you, Dina, for those questions. I think very insightful to have some understanding on the correlation between food safety and muscle myopathies, which is none, but or at least there's no evidence of that. With that, I'm going to take over from Dina and introduce our next speaker. Our next speaker is an associate in the Department of Animal Sciences and he will be talking to us about pre-harvest interventions and implications on safety of turkey processing. With that, I will turn it over to Anup, and I will mute myself and shut my video.
Thanks, Manpreet. So can you hear me clearly, Manpreet?
Yes, I can. And we can see your video also. We can see your cool microphone too.
Thank you. Thank you. Thanks, Manpreet and Harsha, for your great presentations. And good morning, everybody. And as Manpreet pointed out, I'm Dr. Anup Johny, an associate professor of poultry interventional food microbiology and safety in the Department of Animal Science at the University of Minnesota. Thank you, Midwest Poultry Federation, for hosting this convention despite the COVID issue. Thanks, Manpreet, for inviting me to be a presenter in this group. Thanks, Dina, for chairing this session, and good to meet Harsha virtually once again. And today I'm going to talk about pre-harvest interventions and their implications of microbiological safety for turkey processing. And I will be discussing this from a salmonella control angle. So turkey meat is a global product that has a stable demand across the world, especially in the developed countries. And turkey meat consumption is largely determined by the purchasing capacity of the population and seasonality of their use. For example, Thanksgiving, holidays, and Christmas are the seasons where we buy more turkey meat. And thus people who have more purchasing power traditionally have higher per capita consumption of turkey meat. On the other hand, the rise in the living standards and urbanization, increasing popularity of Western diets, and higher attention for lower fat intake have led to a significant increase in the production and consumption of turkey meat across the world over the past decade. So U.S. is the largest producer and exporter of turkeys in the world, implying an economic impact of roughly 110 billion US dollars, offering jobs to around 440,000 people, which is equivalent to approximately 24 billion in wages and approximately 8.5 billion in taxes. So increasing consumer attention toward low-fat, high-protein diets and demand for healthy foods has led to a significant increase in the production and consumption of turkey meat, both in the United States and around the world. So consumption of turkey in the United States is currently accounted at approximately 13 kilograms per capita. So the presence of microorganisms in turkey meat production is a concern to the producers, retailers, and consumers. There are two main categories in which microorganisms of concern can be listed. The first are those that affect the rate of spoilage, and the second, those that present biological risks, resulting in food safety threats to humans. The USDA's Food Safety Inspection Service has indicated that the potential foodborne illness-causing microorganisms associated with turkeys include those of Salmonella species, Campylobacter, Staphylococcus, and Listeria monocytogenes. Now, colonization of live birds and contamination of products with human enteric pathogens present a significant challenge for the industry because poultry, including turkeys and livestock, serve as reservoir hosts for these pathogens. Salmonella and Campylobacter are commonly found in food-producing animals and birds, including turkeys, as we all know about. Both microbes are gram-negative rods, Campylobacter being spirally curved rods, as you see there on the slide. Campylobacter is microaerophilic to anaerobic in its capacity to utilize oxygen, and Salmonella could be aerobic to facultatively anaerobic due to their respiratory adaptations. Now, one of the major food safety threats that the turkey industry faces today is the presence of Salmonella. Salmonella is the leading cause of foodborne illness in the United States, resulting in an estimated 1.4 million foodborne illnesses per year, leading to approximately 26,000 hospitalizations and around 400 deaths. In the recent years, the United States poultry production has encountered severe foodborne outbreak setbacks from various reservoirs of Salmonella, which includes the well-known serovars and the emerging. The common serovars possibly would be S. enteritidis and Salmonella typhimurium. Now, there are quite a few of the serovars that are currently emerging in turkey production, and a significant concern with the emerging serovars is the potential to develop antimicrobial resistance in these strains. So in 2011, a multi-drug resistant serovar, Salmonella Heidelberg, caused an outbreak in the United States, resulting in close to 140 cases of foodborne illnesses across multiple states due to the consumption of contaminated ground turkey. In the same year, an outbreak of MDR of Salmonella Hader was linked to the consumption of contaminated turkey burgers. And additionally, a massive multi-state outbreak of salmonellosis was caused by another serovar, Salmonella Redding, resulting in 356 illness cases across 42 states in the United States. The products implicated in the outbreak includes contaminated raw turkey pet foods and turkey products, as well as live turkeys. So in line with these outbreaks, around 78,000 pounds of raw ground turkey products were recalled due to the contamination with Salmonella schwarzgrund, an emerging foodborne outbreak causing serovar. As of May 2019, this outbreak has resulted in 7 reported cases of salmonellosis across 3 states and resulting in recall of ground turkey products. One of the key problems with Salmonella is that they can be present anywhere along the breeder farm, through the hatchery where the eggs are hatched, to the brooder houses and grow-out houses, processing units, transportation, kitchens, and restaurants. Additionally, so many other sources are present on farms that worsen the situation, which I will discuss in the next slide. So turkeys can serve as natural reservoir hosts for Salmonella, and colonization in birds are often considered to be asymptomatic, and Salmonella therefore can be excreted in the feces and spread to the healthy birds. So control of Salmonella is particularly difficult as there are multiple sources by which the birds are exposed to and potentially colonized by the pathogen, including contaminated feed or water, personnel, or from other birds of the flock. So similar to chickens, Salmonella enters through the mouth and then goes all the way through the intestinal tract and colonize the cecum, and where they will multiply and get excreted, contaminating the farm and the incoming flocks. And we recently found that Salmonella can get disseminated to the liver and spleen once colonized in the cecum, which is what we also find in chickens and layers. So Salmonella's commensal presence in the GI tract of poultry makes it difficult for producers and processors to identify infected birds, and thus complicates the whole effort to prevent the dissemination of Salmonella in the food supply chain. Traditionally, as we all know, 2 intervention approaches are applied to reduce pathogen load to the maximum extent possible. They are the pre-harvest intervention that a producer, breeder, hatcher can employ to reduce the pathogen load. And the post-harvest intervention can be applied anytime during the post-slaughter processing stages that further bring down the pathogen load before it reaches the consumer. And now, although antibiotics were previously used to reduce pathogen load in birds, the advent of a veterinary feed directive issued by the United States Food and Drug Administration has curbed and mandated veterinary oversight on a number and amount of antibiotics that can be used on farms. And this has resulted in the search for alternatives to antibiotics to address the gaps that once antibiotics had. So the common pre-harvest alternatives that could be effectively applied in turkey production include probiotics, prebiotics, vaccination, phytobiotics, organic acids, antimicrobial peptides, bacteriophages, etc. Some of them are currently going through research. Some of our ongoing intramural and federally funded projects investigate the potential of these interventions for applications in poultry, including turkeys. So among these strategies, I'm going to focus on probiotics for this talk. In general, as we all know, probiotics are live microbes, the select species and strains having capabilities to improve the host intestinal microbial balance, improve immunity of the host, produce antimicrobial substances against pathogens, and effectively excluding harmful bacteria from the gut. So a highly effective intervention for application on turkey farms is the use of host-derived or non-host-derived probiotics. We at the University of Minnesota are studying a select few of these two groups of probiotics to strain level for their applications in turkeys and potentially explore their mechanisms of action as observed in turkeys. Two such beneficial microbes whose probiotic qualities and potential to control Salmonella in live turkeys that we are exploring are the Propionibacterium and Lactobacillus species. So to begin with, Propionibacterium fruthenrechi, dairy origin, generally recognized as safe status probiotic, was tested against multi-drug resistant Salmonella Heidelberg in live turkeys at different age groups. The probiotic caused a significant reduction of Salmonella Heidelberg in the cecum of turkeys, irrespective of their age. However, the magnitude of reductions varied among the different age groups. We found that the Propionibacterium supplementation through water resulted in 2-log reduction of Salmonella Heidelberg in 2-week-old turkey pulse, and a reduction of close to 2 logs were found in Propionibacterium fruthenrechi supplemented groups when the study was conducted in 7-week-old grower turkeys as well. Apart from the colonization of Salmonella Heidelberg reduction in the colonization in the cecum, the probiotic supplementation was also effective against the invasion of Salmonella Heidelberg into the liver and spleen without adversely affecting the body weights in turkeys. Now, additionally, we also investigated lactobacillus strains of turkey origin and found to have high probiotic potential against Salmonella Heidelberg. And supplementation of lactobacillus strains through water resulted in a reduction of Salmonella Heidelberg colonization in the cecum of turkey pulse, as well as invasion of spleen and liver. Our ongoing projects with lactobacillus strains reveal that they are equally effective in adult turkeys against Salmonella Heidelberg. Now, it is understood that probiotics could result in reduction in the cecal pH because of the released lactic acid and peroxides and other compounds that would itself be inhibitory on gut pathogens. In addition, the antimicrobial function of probiotics could be through exercising competitive exclusion, nutrient competition, production of metabolites such as antimicrobials, stimulation of immune cells, and regulation of tight junctions. Given our promising finding that the dairy probiotic is highly effective against Salmonella, our approach was more geared towards understanding the microbiome structure as to how the pathogen would be shifting the bacterial population in the gut while they colonize, and the effect of dairy probiotic in restoring or balancing the gut health imprints. And this is particularly important since we don't have much targeted studies to understand the microbiome signature of Salmonella colonization and restoration by probiotics from an interventional food safety site. And this research has been supported by Minnesota State Legislature funds, University of Minnesota, and the USDA. Now, for the microbiome analysis, we collected cecal samples from the turkeys infected with drug-resistant Salmonella Heidelberg, with or without treated with Propionibacterium, and conducted an Illumina 16x sequencing, and appropriate interpretations were made. We tested it at 2 weeks, 6 weeks, 7 weeks, 11 weeks, and 12 weeks. Overall, as you see in that figure, that represents the effect of Propionibacterium on the major bacterial phyla. We found that Propionibacterium was supporting gut health and assisting in the restoration process as a result of Salmonella colonization. Firmicutes, Tenoricutes, Bacteroidetes, the common and major phyla in the gut were the abundant phyla, but their compositions changed as their age advanced in response to Salmonella challenge, indicating that the colonization of Salmonella produces changes in the microbiome and that the dairy probiotics are restoring them by supplementation at a broader bacterial population level. Now, it's much clearer in these figures as well. The left figure is indicative of the Shannon index that depicts the alpha diversity among the treatments. The alpha diversity provides an indication of species richness and abundance. The more, the better. You can see that even though there were differences in the species genera, changes in each treatment as we saw in the previous figure, in the previous slide, the gross abundance and richness did not change, which is very unique and probably a newer finding in relation to Salmonella challenge and probiotic supplementation studies. And on the right panel, you can see that the beta diversity, which is represented in a principal component analysis plot, and you can see that there is an apparent clustering of the samples based on treatments, the red being Salmonella, blue being negative control, where no treatments are applied, and green being the probiotic supplementation. So the beta diversity is the difference in the bacterial composition across different samples. And apparent clustering indicates differences in the bacterial composition in response to different treatments. So taken together, the two slides, what we find is that Salmonella, being a great colonizer without showing any obvious clinical signs, did not cause a significant change in the broader bacterial population, but on a deeper analysis, indicated abundance of specific indicator bacterial species. And likewise, we observed that Propionibacterium restored the lost balance of microbiome in the gut, but causing the abundance of yet another group of indicator bacteria when they were supplemented. And this shows how the bacterial realm is dynamic in its response to the infection process and probiotic treatment. For more information on this, please contact me. I would be glad to discuss. So that is nice. So probiotics work against Salmonella. So are some of the other interventions. They do a significant job in reducing Salmonella. But will that safe level brought in by these pre-harvest interventions enough for a final safer product at the consumer's kitchen? Remember that our hurdles are continuing during the processing steps as well. We must understand how pathogens like Salmonella move through the processing steps before trying to approach this question. Now, this is what's happening in the processing line. A detailed processing step has been already discussed by Manpreet and Harsha. Turkeys will be received and slaughtered in the processing plants, and the carcasses move along the cleaning, evisceration, processing, preparation, packaging, and shipping steps until it reaches the retailer and then to the kitchen of the consumer. Like pre-harvest sources of Salmonella in a farm, several opportunities exist for the pathogen to be present in the processing plant and get the carcasses contaminated. So due to high sanitation methods and inspection processes that go on in the processing line and less time elapsed during the whole process, the source of contamination could be limited to a few. So this slide shows a close view of the processing steps. The birds are stunned, blood, and then go through the scalding tank. The scalding step, where carcasses are immersed in large pools of hot water, is a critical control point, as this creates the potential for transfer of bacteria between carcasses at the beginning of processing events. And after scalding, the feathers are picked, internal contents removed, and the carcasses are washed and chilled. As Manpreet pointed out, picking or evisceration steps are contamination points. Although there is less probability for pathogens to remain viable in the chiller tanks, they can also serve as cross-contamination points. Therefore, although we take care of majority of the pathogens prior to slaughter, we have to take care of the scalding and chilling stages and other cross-contamination stages due to the potential for cross-contamination. So one of the key aspects of Salmonella is its ability to attach to poultry skin. In both scalding and chilling tanks, Salmonella can attach strongly onto the skin. In addition, the oiliness of the skin facilitates the pathogen attachment, and the organic matter in the tanks reduces the efficacy of disinfectants. So these are some of the post-harvest interventions that include physical, chemical, and biological. Some of the examples are electrolyzed oxidizing water, high-pressure inactivation, organic acids and oxidizers, peracetic acid, and the emerging group of bacteriophages. So peracetic acid is common in turkey processing, as it is an approved antimicrobial available as peracetic acid hydrogen peroxide mixture. And studies indicate that approximately 100-fold reductions or more of bacteria could be achieved using the compound. And we are currently investigating peracetic acid in our studies in detail, as we have some comparable results with it as well. So what we are focusing on is plant-based options at these stages. We are exploring the essential oils capability as alternatives for processing aids in scalding and chilling, and as post-chilling dip treatments. These include trans-cinnamaldehyde from cinnamon, carbachol and thymol from oregano, bitter salicylic acid from flowering plants like angiosperms, lemongrass from lemongrass essential oil, and pimenta from allspice. So what we have found so far is that pimenta from allspice essential oil and lemongrass from lemongrass essential oil are highly effective against Salmonella heidelberg on turkey drumstick skins and in processing waters. Yes, scalding step is difficult for antimicrobial interventions to work due to the high organic content and the possibility of antimicrobials being neutralized over time and in the presence of organic matter, as Manpreet pointed out. However, we believe that scalding step should be addressed for improving the food safety of turkey products as well. We also found that cinnamaldehyde and lemongrass reduce the Salmonella heidelberg in ground turkey patties during refrigerated storage for 5 days. Sorry, the slide actually moved. So I wanted to talk to you that if you need more information on essential oil application in poultry, please watch the extension YouTube video where I expand on essential oils, their mechanism, and our group's actively ongoing research. And this slide I've already discussed. As we found the essential, the efficacy of pimenta essential oil and lemongrass essential oil and trans-cinnamaldehyde on ground turkey patties and their efficacy in scalding and chilling waters. Now, moving forward, we all understand that Salmonella enters the production and processing at multiple points. Given our studies so far, we can support the thought that pre-harvest interventions are essential as they reduce the initial pathogen load in turkeys before they reach processing plants. This will alleviate the burden on the current post-harvest control systems employed by the industry. And we are currently working towards exploring the potential of these interventions against the emerging Salmonella in turkey industry, such as Salmonella redding. So I would like to acknowledge my team members who worked and are working on these projects, Drs. Divek and Luna, and my doctoral students, Grace, Claire, and Shigina, and my former lab manager, Jason. I do want to thank the funders of the research as well, the USDA AFRI Mitigation Strategies Program, where I collaborate with Drs. Tim Johnson, Sally Knoll, and Carol Cardona, the USDA Organic Program on Broiler Processing, where I collaborate with Dr. Annie Donahue at the USDA ARS Arkansas, and Kumar Venkatnarayanan at UConn, Minnesota Rapid Response Funds, and the USDA Hatch Funds. The Minnesota turkey growers have been my stakeholders, and their funds are highly appreciated, specifically the support from Dr. Petri Papinaho of Genio Turkey Store. And I would also like to thank the Immune Drive and the USDA National Needs Fellowships awarded to my doctoral students who took time to work on the manuscript draft and their collaborative efforts in the research. And thank you, everyone, for your patient hearing. Thanks, Dina, Manpreet, and Harsha for this memorable presentation even. I'll take any questions that you may have now. Thank you.
Thank you, Anup, for a wonderful presentation. I do have one question, if I can start off. You talked about the essential oils showing promise against pathogens. Do you have a sense of the cost of those and the amount that you would need to generate for effectiveness against pathogens?
That is a great question. And we are currently doing our work with North Carolina State University on the cost-benefit analysis. The cost at this point with respect to some of the essential oils which are emerging, for example, pimenta and lemongrass, are a bit expensive. They are expensive at this point, but we have other essential oils that are bulk marketed, and we have tested the concentrations which are high at this point, but we have found compounds that can be used to dilute them, to solubilize them in the water, especially in the scalding and chilling tanks. And currently we are working with Tom Bukina at North Carolina State in terms of getting the economic analysis done. So we are hoping that we will have some of the preliminary findings out as a part of that USDA organic grant program. Thank you. That's a great question.
Okay. Wonderful. I'm going to call in Dina and Harsha. Also, I don't see any other questions in the chat window. Anything else? Harsha, do you have anything, or Dina?
No, I don't. Manpreet?
Okay. I think with that, we just have a couple more minutes before we end. So I want to take the time to thank Anup and Harsha for accepting our invitation to talk about their experiences and providing us insights on the issues that are very relevant to the industry. And we hope that you guys will enjoy the rest of the sessions in the virtual conference. With that, I thank you all for attending. And yes.
I'm going to add in a little something. It's also the attendees will notice the sponsors on towards the bottom side of the screen. Those are the sponsors are allowing or have sponsored to allow for these sessions to be available through the end of the month through the site. So just to acknowledge their participation in the program also.
Wonderful. Thank you, Dina, for keeping me in line. So now with that, I thank you all for your attendance, and I hope you enjoy the other sessions, and hopefully we'll get to interact with you on some of the other forums. Thank you.
Thanks, Manpreet. Thank you.
Thank you, Manpreet.